Hello, cellular and molecular biologists. Today, Kevin, Sydney, and I are going to be talking to you guys about CapG as an oncogene and its involvement in the migration and invasiveness in ovarian carcinomas. Before we get into the experiment itself, we want to go over why this research is important. Ovarian cancer is the fifth leading cause of cancer-related death among women, with the average 5-year overall survival of advanced stages not exceeding 40%. The tumors involved in ovarian cancer can be classified as type 1 or type 2. Type 1 tumors are slow-growing and develop from precursor lesions. Type 2 tumors, on the other hand, display metastatic activity and are much more fatal. Mutational analysis revealed that 50 to 80% of these tumors harbor various protein 53 mutations, and while they show genetic instability, certain sequences causing these tumors have not yet been identified. To better study how these tumors migrate and invade into other parts of the body, researchers studied CapG. CapG is recognized as an oncogene and is associated with invasiveness in breast cancer. CapG stands for capping actin protein. This protein regulates actin polymerization by binding to the positive end of the actin, preventing the addition of subunits. This allows for disassembly of the negative end of the actin, and once enough actin subunits are released and processed, the capping protein is removed from the plus end and growth can continue. CapG overall promotes cell motility and regulates the rate and site of actin polymerization. Single nucleotide polymorphism Rs6886 affects CapG translation and consequently its expression. In this SNP, the G nucleotide is replaced with an A. It affects the phosphorylation of serine by destroying the protein kinase C recognition motif. For this experiment, the presence of SNP Rs6886 will be compared in individuals with ovarian cancer as well as healthy controls. The purpose of this experiment is to determine the impact of CapG on migration and invasiveness in ovarian cancer cell lines. This study hypothesizes that metastatic ovarian cancer cell lines display increased expression of CapG. CapG expression will be measured among several other ovarian cancer cell lines using real-time PCR. Additionally, a single nucleotide polymorphism located in exon 10 of the CapG gene will be analyzed to determine if it can be linked to fallopian tube and ovarian carcinomas.
Multiple experiments were carried out in this paper, but to start, the researchers obtained ovarian cancer and adjacent tissue samples. They also took different cancer cell lines including SKOV3, OVCA3, HEY, and MDAH cell lines. These samples were ran through reverse transcription qPCR in order to find the levels of CapG gene expression. The HEY cell line has a naturally low level of CapG expression, so in order to study the effects of CapG, HEY was modified to overexpress CapG. To do this, human CapG was amplified with PCR, was then digested and cloned into S11IN, which is a retroviral expression vector. The vector was then transfected into HEY cells in order to create the mutant HEY CapG. Where HEY cells had a low level of CapG expression, SKOV3 cells had a high expression of CapG. A mutant was created that had the CapG gene knocked out using siRNA, or small interfering RNA. The siRNA has affinity for the coding region of CapG, and when bound, cleavage is induced where the siRNA is bound. The knockout cells that result from the siRNA transfection are referred to as SKOV3 plus siCapG. After the creation of cells with knockout and overexpressed CapG, a scratch assay was performed. A scratch assay measures the amount of time it takes for the samples to heal after being cut. To make the scratch, a pipette was used to create 3 vertical scratches for each cell line. They waited until the first wound appeared to be closing and then used methanol to freeze the cells in place. These snapshots were then measured. They compared the movement of SKOV3 and the CapG knockout version, and also the HEY and the overexpressed CapG version of HEY. Next, to measure how cancer cells metastasize, an invasion assay was performed. The invasion assay measures what proportion of cells move from one chamber to the next over a period of time. First, the cells were put into the top chamber in blue. If the cells are invasive, they will move from the first chamber into the second chamber, which is purple. For the experiment, after 22 hours, the lower chamber was removed and the membrane was cut into a slide, which was then observed. To confirm the knockout and overexpressed versions and the CapG expression of all variants, SDS page gels and Western blots were performed to confirm levels of the CapG protein. As a side experiment, 263 ovarian cancer patient DNA samples and 107 control DNA samples were analyzed using HRM, or high-resolution melting. Each nucleotide has a different melting point, so as the CapG gene is ran through HRM analysis, mutations are able to be found by comparing the melting curve of the patient with the curve expected from a normal genotype.
Now for the results of this research. First, the authors wanted to measure CapG expression in ovarian carcinoma tissue and in the ovarian carcinoma cell lines. To do this, they utilized RT-qPCR as mentioned previously. The results are displayed in this figure, where on the y-axis you can see relative CapG expression normalized to a housekeeping gene, GAPDH. On the x-axis, we have normal tissue and tumor tissue. The average expression in normal tissue was utilized as a cutoff to indicate overexpression, which in this case is anything over 2, and can be seen here by this dashed line. Therefore, you can see that about 38% of the tumor tissue samples had CapG overexpression. Additionally, CapG expression of the 3 ovarian carcinoma cell lines was measured. The Hay cell line contained the lowest CapG expression, which was below the cutoff, while the SKOV3 and OVCA3 cell lines exceeded normal expression. All 3 cell lines had CapG expression that was in the same range as the OC tissue. The authors also wanted to estimate the impact of CapG expression on cell migration in ovarian carcinoma cell lines. In order to do this, they needed to utilize the same OC cell lines, but with modified CapG expression. The first cell line they used was SKOV3, which has high CapG expression, and was utilized to create the cell line SKOV3 SI-CapG that has decreased CapG expression. The Hay cell line was also used, and this cell line contains intrinsically low CapG expression, so it was utilized to create a cell line with CapG overexpression, known as Hay CapG. RT-qPCR was then utilized to measure the relative expression of CapG in each of the unmodified cell lines and the 2 new modified cell lines. SKOV3 SI-CapG was confirmed to have a 4-fold reduction in CapG expression compared to the original cell line. The Hay CapG cell line was then confirmed to have a 230-fold higher expression of CapG when compared to the original Hay cell line. CapG protein expression was also measured with the Western blot analysis. Here at the top we have our 2 original cell lines and our 2 modified cell lines. CapG silencing can be confirmed with the lack of band here, while CapG overexpression in the modified Hay cell line can be confirmed with this larger band present here. Additionally, alpha-tubulin was utilized as a control, confirming equal protein quantities in each sample. After creating these modified cell lines, the authors utilized a scratch assay in order to measure cell migration rate. First, let's look at the SKOV3 cell line compared to the CapG knockout cell line. The mobility of the CapG silenced cell line was nearly half that of the original cell line. This decreased mobility of the CapG knockout can be seen here. Next, you can see that the cell line overexpressing CapG had a significantly higher migration rate when compared to the original Hay line. This increased mobility can be seen here clearly after 72 hours. Based on this, the authors concluded that CapG expression levels were positively correlated with migration potential. Invasiveness was also measured in the SKOV3 cell line following CapG knockdown with the matrigel transwell invasion assay. After 24 hours, the knockdown cell line was determined to have a significant decrease in invasiveness compared to the unmodified cell line. This is visually represented here, where you can see that a smaller amount of the CapG silenced cells were able to migrate across a basement membrane matrix. This confirmed that an increase in CapG expression resulted in an increase in cell invasiveness. Additionally, the authors wanted to analyze post-translational modifications of the CapG protein, such as phosphorylation. One such phosphorylation site was identified to be a serine at position 337. It was determined that this phosphorylation is affected by SNP RS6886. This single nucleotide difference ultimately resulted in an arginine being exchanged for histidine in the CapG protein, which prevented its phosphorylation. Next, an association study was carried out to analyze allele frequencies for this particular SNP, in order to analyze its impact on various cancers. This was done by genotyping DNA samples from both healthy patients and those with ovarian or fallopian tube cancer. Here on the left, we have the different genetic variants, which are homozygous for histidine, homozygous for arginine, or heterozygous. And then here we have the frequency of these different genetic variants in the healthy group. Next is the frequency of these variants in a population of patients with ovarian carcinoma and patients with fallopian tube carcinoma. You can see there was a difference in the frequency of the homozygous A genotype in the OC patients when compared to the control. However, this was not significant. However, there was a significant difference between the fallopian tube carcinoma group, which had 50% of the population contained this homozygous A genotype compared to the 10% in the control. Therefore, the authors could conclude that SNP RS6886 is linked to fallopian tube carcinoma. However, further research is needed. Now to summarize these results. So first, the authors concluded that CapG is overexpressed in ovarian carcinoma tissue. Also, that these ovarian carcinoma cell lines show CapG-dependent migration and invasiveness. They also discovered that the SNP RS6886 affects CapG phosphorylation and potentially modifies its function. And finally, that this SNP may be correlated with the development of fallopian tube cancer. And that's it. Thanks for watching.